You might be asking yourself what purpose there would be in generating an inhibitory postsynaptic potential in the chain of cells that leads either to the central nervous system or away from the central nervous system. What would the purpose be in making the postsynaptic membrane harder to depolarize? Well, this slide gives us the answer. So far, we've been talking about neurons as though one neuron communicates with another neuron, and we have this one-to-one interaction. But in reality, neurons talk to a lot of other neurons all the time. Not just neurons, but remember there are other effector cells. There's muscle and glands involved as well. The result is what you're looking at on this slide and what you're looking at over here in this scanning electron microscope image. In this drawing, we're looking at the cell body of a neuron right here, and we're looking at a whole bunch of dendrites both coming off of that cell body and also coming in and synapsing with that cell body. You can see the axon heading out here. It happens to be a myelinated axon. Here's the axon hillock. So everywhere you see a little green dot, that represents a synapse. And everywhere you see a little red dot, that represents a synapse. And these all happen to be chemical synapses involving neurotransmitters. Everywhere that there's green, the neurotransmitter is binding to the postsynaptic membrane and generating an excitatory response, an EPSP. Everywhere you see a red dot, we've got IPSPs being generated. Now, what does this neuron do? What does this neuron do when it's being stimulated in two completely different ways by a whole bunch of other neurons? Well, the answer is it sums up all of this input. So the sum of all of the excitatory synapses and the inhibitory synapses together is going to cause information to be sent along to the next cell. The sum is what matters. Each and every neuron in your nervous system is receiving an abundance of information at any one time. Not just one stimulus, but many, many stimuli. And when those stimuli come in the form of neurotransmitters in chemical synapses, it creates a whole lot of information that then impacts what information gets sent along. So it's not a one-to-one. It's a summation going on. Each one of these little synapses, each place where one cell is communicating with this cell, there's either an EPSP or an IPSP being generated, and this cell is going to take all of that together and send that information along down its axon. So the next thing I'd like to talk about are these neurotransmitters, these signaling molecules that are released and travel across chemical synapses to elicit a response. As you probably know, there are many different types of neurotransmitters, and these different neurotransmitters are going to be released at different times and under different circumstances in order to create the very complex range of responses that our nervous system is capable of. The critical thing about neurotransmitters is that they have to elicit responses once they are released. So cells have to keep very tight control over when a neurotransmitter is released and how much of it is released, and how long it's allowed to float across that chemical synapse and interact with the next cell. Cells keep a very tight rein on neurotransmitters because they are so powerful. So we're not going to have an opportunity to talk about all of the different types of neurotransmitters, but we will discuss 4 important groups of neurotransmitters. The first one is a single neurotransmitter called acetylcholine. The second one is a group of neurotransmitters called catecholamines. And this group includes things like dopamine, epinephrine, and norepinephrine. Third is a group called the amino acids and the amino acid derivatives. And this includes things like histamine and serotonin. And finally, there's a group called the neuropeptides. And the neuropeptides include the enkephalins. On this slide, what you're looking at up at the top, if I can enlarge it for us here, is the synthesis reaction that produces the neurotransmitter called acetylcholine or acetylcholine. Up at the top, you can see this molecule acetyl-CoA. You've seen this before. You've seen this molecule during the process of glucose oxidation. Remember, acetylcholine comes out of pyruvate processing and is fed into the Krebs cycle. So during this synthesis reaction, acetyl-CoA and choline come together. The CoA portion of the molecule is released, and an enzyme called choline acetyltransferase combines the substrates to produce acetylcholine. Again, this is an important enough neurotransmitter that we're going to say a few things about it in just a minute. What I'd like us to be familiar with regarding the catecholamines is that they are all structurally related to each other, and that makes sense. If you look at this reaction here, it's showing us that the molecule called dopamine is used to build the molecule called norepinephrine, and from norepinephrine can be synthesized epinephrine. So not only are these 3 in the same family of neurotransmitters, but they are synthesized from one another. And finally, down at the bottom here, they're just showing us a couple of the amino acid derivatives. Again, things like serotonin, histamine. These are important neurotransmitters as well. So I want to say just a few things about acetylcholine. Acetylcholine is the most common neurotransmitter in vertebrate animals in terms of the peripheral nervous system. So not just human beings, but all vertebrate animals. It's the most common neurotransmitter out in the peripheral nervous system, and we primarily think about it in relation to the neuromuscular junction. This is the neurotransmitter that gets released at the junction between the nervous system and the skeletal muscles. Acetylcholine is an excitatory neurotransmitter, so when it is released into a chemical synapse and binds to receptors on the postsynaptic cell, it's going to elicit an excitatory postsynaptic potential. It's going to stimulate, in other words, action potential generation on the postsynaptic side. Specifically, when acetylcholine binds, it's to a receptor that acts as a sodium channel. So the binding action to that receptor channel is going to cause the channel to change shape and open up, allowing sodium to flow in to that postsynaptic cell. Any synapse in the body that uses acetylcholine as its neurotransmitter gets a name. We call these cholinergic synapses. And again, they're quite abundant in the body because of the abundance of acetylcholine. The catecholamine group, remember, includes dopamine, norepinephrine, and epinephrine. Now, this group of neurotransmitters is a little more complex in its effects. Depending on the synapse, they can be either excitatory or inhibitory in their action. You find catecholamines in chemical synapses between neurons, so neuron to neuron. They're also quite abundant between neurons and smooth muscle cells, in the intestines particularly. And they're also quite abundant in neuron to neuron synapses in the brain. Any synapse that uses a catecholamine neurotransmitter gets a name. We call those adrenergic synapses. The amino acids and amino acid derivatives include, again, histamine, serotonin, a derivative called gamma aminobutyric acid, or GABA. Glycine is one, glutamate is one. These are just amino acids that are capable of acting as neurotransmitters. And like the catecholamines, the amino acids and the amino acid derivatives can be very complex in their action. It depends on the synapse that they're involved in, whether they're going to elicit excitatory effects on the postsynaptic cell or inhibitory effects. The neuropeptides are probably one of the most interesting types of neurotransmitter and probably the least understood. Like the name suggests, these are peptides. These are short chains of amino acids that function as neurotransmitters. And this is a big group of neurotransmitters. Hundreds of neuropeptides have been identified so far, and it's believed that there are many, many more that have just not been discovered yet. These are a different type of neurotransmitter compared to the other ones we've discussed so far, because rather than being released and acting on one postsynaptic cell, the neuropeptides tend to be released in areas where groups of postsynaptic cells are involved in a synapse. So the effects are going to involve multiple postsynaptic membranes. Neuropeptides tend to have long-lasting effects. And depending on the neuropeptide that you're talking about, some of them are excitatory, some of them are inhibitory on the postsynaptic cell. A couple of examples of neuropeptides. There's one called substance P that is excitatory. And there's a group of neuropeptides referred to as the enkephalins. This is a fascinating group. These have inhibitory effects. The enkephalins are probably best known for their role during episodes of shock and extreme stress. So imagine, for example, stories you may have heard of people who have been badly burned in some sort of accident. There are stories that first responders will tell of burned people, badly burned people, just walking away from an accident site, walking out of a burning building, or walking away from a car accident, and talking and acting like nothing's wrong. And the reason they're able to do that is because they are in such shock in that moment. They are at a level of such extreme stress. Their brain is flooded with enkephalins, and all of the sorts of stress-related, pain-related reactions that they might otherwise have are being inhibited. This kind of inhibition may well be an evolutionary survival reaction, right? A person who has been badly burned in a fire needs to get up and get out of the fire. So it may be an evolutionary process that temporarily allows that person to remove themselves from the danger that they're in. Neuropeptides are relatively long-lasting compared to other neurotransmitters, but this kind of extreme response that we see with enkephalins, it's not going to last very long. We're talking about maybe minutes, but not super long, not hours and hours and hours. Very, very interesting group of neurotransmitters. We said earlier that neurotransmitters are manufactured up inside the neuron cell body, just like all other materials are manufactured up there. And they are packaged into secretory vesicles, and they are sent down along microtubule tracks down the length of the axon using motor proteins so that the vesicles can arrive at the terminal branches and the terminal bulbs, or synaptic knobs as they're called, and then stay down there, get stored down there in these transport vesicles. The correct time and under the correct circumstances, the neurotransmitter will then be released into the chemical synapse. Remember too that we said that neurotransmitters have to be kept under very tight control by the cell because they are so powerful. And part of the way that the cell controls neurotransmitters is by controlling their release. And that is done with calcium. Now, we've seen calcium play a role like this before. We saw calcium act as a second messenger during the process of exocytosis. And neurotransmitter release is essentially exocytosis. So it makes sense that calcium's involved. The difference here is that rather than having a hormone bind to a receptor and trigger calcium release, this time it's the arrival of the action potential. And that action potential, as it comes down the terminal branch and hits that bulb or knob, channels, calcium channels in the membrane are going to open up. These are voltage controlled calcium channels. And as calcium flows in, it's going to increase the concentration of calcium in that terminal bulb. Remember that the calcium is going to help activate protein kinase enzymes. Protein kinase enzymes are going to phosphorylate proteins on the outer layer of the transport vesicle membrane. That's going to allow the vesicles to fuse with the presynaptic membrane. Remember, proteins on the outside of the vesicle get phosphorylated. Proteins on the inner monolayer of the cell membrane get phosphorylated, and that allows those two membranes to fuse. There's actually a family of proteins involved in this type of exocytosis, this exocytosis of neurotransmitters. These proteins are called snare proteins. I think that's a great name because these proteins have to grab onto each other. The proteins on the outer layer of the transport vesicle and the inner layer of the cell membrane have to snare each other. They have to grab each other so that fusion of those two membranes can occur and the neurotransmitter can get released out into the synaptic cleft. So cells have control here. It's interesting that neurons that undergo prolonged stimulation, so multiple action potentials coming down that terminal branch, will lead to more and more and more vesicles fusing with the presynaptic membrane. So more and more and more neurotransmitter being released. So cells have control over when they release neurotransmitter, how much neurotransmitter gets released, and then how long it's allowed to stay in the cleft. Remember, neurotransmitters diffuse across the cleft, and once they've been released, they're going to have an effect. So they bind to postsynaptic membrane receptors and, of course, alter the flow of ions in that postsynaptic cell. Cells keep tight rein over this process. On this slide, we're looking at this process that we just talked through in a diagram. Right here, you're looking at the terminal branch of an axon and the bulb at the end of it, or the knob at the end of it. Over here, you're looking at a postsynaptic cell. Remember, this could be the dendrite of a neuron. It could be a muscle cell. It could be a gland. It depends on where we are in the nervous system. So they're showing us an action potential traveling down and arriving in the terminal bulb. They're showing us this storage of transport vesicles that are all full of neurotransmitter. They are stored down in these bulbs. And as this action potential arrives, and remember it's traveling down the membrane, it's going to open up the voltage controlled gate in calcium channels. And once those channels are open, calcium will be able to flow in to these synaptic knobs. And as calcium levels get higher in here, they will activate protein kinase enzymes, which will phosphorylate proteins on the vesicles and on the inner surface of the membrane, allowing them to fuse. Once that fusion occurs, then we see release. And that's what they're showing us in this diagram. You can see that the vesicle has fused and ruptured, and the neurotransmitter has been released into the cleft. It will interact with and bind to receptors on this postsynaptic cell. Now, it depends, of course, whether or not these receptors are the excitatory type or the inhibitory type. Whether or not we're going to see action potentials regenerated over here or not. If the neurotransmitter is acetylcholine, as we said earlier, we know that action potentials will be regenerated on this side because acetylcholine is an excitatory neurotransmitter. If this is another kind of neurotransmitter, we may or may not see action potentials generated. We're either going to get a depolarization or we're going to have a hyperpolarization. It depends. It depends on the neurotransmitter. It depends on where we are in the nervous system and where this synapse is located. Now, the other place that a cell can control these very powerful neurotransmitters is in the cleft itself. The amount of time that a neurotransmitter is allowed to stay in the cleft is a place where a cell can elicit some control. And most of the time, neurotransmitters are only allowed to act for a very short amount of time before they are inactivated. Now, there's a couple of ways that a cell can do this. A couple of ways that a cell can remove the influence of a neurotransmitter after a short amount of time. Number one, the neurotransmitter can be degraded. Enzymes can be used in the synaptic cleft to degrade the neurotransmitter very quickly. Number two, the neurotransmitter can be taken back up into the presynaptic neuron. And this is achieved through membrane pumps. So a cell wants to control how long it allows a neurotransmitter to act on the postsynaptic membrane. It has a vested interest in controlling the neurotransmitter because it doesn't want it to overstimulate that postsynaptic cell, especially if it's a postsynaptic neuron. So let's start first with this first type of control, degrading the neurotransmitter with enzymes. A good example of this involves those cholinergic synapses, the synapses that use acetylcholine as a neurotransmitter. There's an enzyme called acetylcholine esterase, and this enzyme can break down acetylcholine. We've learned a lot about acetylcholinesterase because there are lots of chemicals in the world that inhibit this enzyme. There are lots and lots of pesticides and herbicides and nerve gases that target this enzyme, acetylcholinesterase. So all of these chemicals, and generally we refer to these as organophosphates. What these chemicals do is inhibit the action of acetylcholine esterase, and that results in acetylcholine being allowed to stay in the synapse and act on the postsynaptic membrane for long periods of time. So you get what are called cholinergic effects, which include overstimulation of the muscles. And people who are poisoned with organophosphate chemicals go into a terrible state where they are in this overstimulated muscle reaction, muscle contraction. People have suffocated and died after being exposed to these organophosphates accidentally. And of course, unfortunately, countries have used nerve gases, organophosphate nerve gases, on purpose. Just recently in the news, there was a story about one of Vladimir Putin's political rivals getting poisoned with one of the organophosphate nerve gases. This man was on his way to Europe on an airplane, and it's believed that a Russian agent slipped one of these agents into his tea. He drank it, and by the time the plane took off, he was in agony. He was basically suffering from over contraction of all of his muscles. He had difficulty breathing, of course. He was rushed to a hospital. He survived. Very, very lucky man, because these nerve gases are very powerful. So, systems are in place in the body to degrade neurotransmitters under normal conditions. Now, on the reuptake side, a really good example of this one is what are called the selective serotonin reuptake inhibitors. You've probably heard of these. These are the SSRIs. This is a class of antidepressant drug. So these drugs have names like Zoloft and Paxil and Lexapro and the granddaddy is Prozac. These are drugs that are believed to act by preventing the reuptake of serotonin in synapses where serotonin is used as the neurotransmitter. There's a theory, and it's still only a theory, that a lack of serotonin or a low level of serotonin in certain synapses is one of the contributing factors to certain mental health problems, including depression. So the idea is, if there's not enough serotonin being released into the synapse, one of the ways we can treat that is by allowing that low level of serotonin to at least stay in the synapse longer. And that's achieved by inhibiting its reuptake. So normally, serotonin gets pumped out of the synapse very quickly and back into the presynaptic neuron. When a person takes an SSRI, that reuptake process is at least partially inhibited. It's not completely inhibited. The serotonin is allowed to stay in the synapse longer and perform its action on the postsynaptic cell for longer periods of time. So, two mechanisms to control how long neurotransmitters are allowed to act, and two examples of how those mechanisms can be inhibited. I want to finish up the lecture today by just bringing this image back up, this image of a single neuron cell body being acted upon by multiple other neurons. I want to remind us that what goes on in the nervous system in terms of communication between neurons and communication between neurons and other cells, muscle cells, gland cells, it's a very complex process. It's not a one-to-one proposition. So there are lots and lots and lots of synapses taking place on every neuron in the system. And all of these synapses are using different neurotransmitters, and different neurotransmitters are having different effects on the neuron. And it's the neuron that then has to sort of sum everything up and send along that summated information along its axon to the next cell. It's a very complex, interconnected system.